Hello, dear students. Greetings to all. I hope all of you are safe. Be safe. Stay at home. Take care yourself and also your family. Okay. So let me share about the enzymology of the genetic engineering. So the enzymes used in the genetic engineering and what are the importance of the genetic engineering. So in the last class we are discussed about introduction of biotechnology, genetic engineering in details. Now in this slide, I am going to explain enzymes of genetic engineering. So, all of us know that enzymes are nothing but the proteins that act as the biological catalyst. So, that catalyst accelerate the chemical reactions, isn't it? So, some of the important enzymes used in the genetic engineering are restriction enzyme, DNA ligase or the ligase, polymerase enzyme, alkaline phosphatase, and the reverse transcriptase. So these are the five enzymes. Those are mostly or popularly used in the DNA or the genetic engineering technology or recombinant DNA technology. So these are the five important enzymes. Okay. So cutting and pasting are two of the first skill children learn, isn't it? And the tools they use are scissor and the glue. Similarly, cutting DNA and pasting DNA fragments together typically are among the first technique learned in the molecular biology lab and are fundamental to all recombinant DNA work. So, genetic engineering became possible with the discovery of mainly two types of enzyme. The cutting enzymes called the restriction endonucleases or the restriction enzymes or the molecular scissor. Okay, and the joining enzymes are called as the ligase enzymes. Restriction endonucleases or restriction enzyme as they are called popularly and recognize unique base sequence, unique base sequence modify in a DNA strand and cleave the backbone of molecule at a place within or at some distance from the recognition site, whereas ligase is the enzyme that joins 5' end of DNA with a 3' end of the same or another strand. So identify of the restriction enzyme. So ಹೇಗೆ ಐಡೆಂಟಿಫೈ ಮಾಡೋದು ಅಂತ? How can we identify the restriction enzyme? Restriction enzymes are named for the organism from which they were first isolated. For example, E. coli, capital R and the I, is isolated from E. coli strain RY13. Okay, isolated from E. coli strain yadu antandre RY13. Eco refers to the genus and species. First letter of genus, first two letters of specific epithet. R is the strain of E. coli. I, Roman numerical, indicates it was the first enzyme of that type isolated from E. coli RY13. ಹಾಗೆ so same BAM or the BAM ಅಂತ ಕರೀತಾರೆ. It is called as the BAM. BAM H first is isolated from Bacillus aeruginosa strain H. SA3A is isolated from Staphylococcus aureus strain 3A and so on. Some restriction enzymes also cut DNA from blunt ends, means without single stranded tails, which also can be inserted into target DNA via the action of DNA ligase. Restriction enzymes are found in bacteria and archaea and provide a defense mechanism against invading viruses. So, explanation of the restriction enzymes are: these are the enzymes that cut DNA at or near specific recognition nucleotide sequences known as restriction sites. ಓಕೆ, ಈ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಏನು ಮಾಡುತ್ತೆ ಅಂತ ಅಂದರೆ ಡಿ ಎನ್ ಎನ ಕಟ್ ಮಾಡುತ್ತೆ. ನಿಯರ್ ಆಗಿರ್ಬೋದು ಅಥವಾ ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಸೈಟ್ ಅಂತ ಇರುತ್ತೆ, ಅದನ್ನ ಕಟ್ ಮಾಡುತ್ತೆ. ಅದು ಯಾವ್ದು ಪ್ಲೇಸನ್ನ ಕಟ್ ಮಾಡುತ್ತೆ, ದಟ್ ಸೈಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ಸ್. The restriction enzymes or that cut DNA at or near specific recognition nucleotide sequences known as the restriction sites. Restriction sites. Isolated restriction enzymes are used to manipulate DNA for different scientific applications and are an important tool for recombinant DNA technology. Restriction enzymes, also called restriction endonucleases, a protein produced by bacteria that leaves DNA at specific sites along a molecule. In the bacteria cell, restriction enzymes cleave foreign DNA, thus eliminating infecting organisms. Restriction enzymes can be isolated from bacterial cells and used in the laboratory to manipulate fragments of DNA such as those that contain genes. For this reason, they are indispensable tools of recombinant DNA technology or the genetic engineering. A bacterium uses a restriction enzyme to define against bacterial viruses called the bacteriophages or the phages. When a phage infects a bacterium, it inserts its DNA into the bacterial cell so that it might be replicated. The restriction enzymes prevents replication of the phage DNA by cutting it into many pieces. So restriction enzymes were named for their ability to restrict or limit the number of strands of bacteriophage that can infect a bacterium. Each restriction enzyme recognizes a short specific sequences of nucleotide bases, the four basic chemical subunits of the linear double stranded DNA molecule. What are those? Adenine, cytosine, thymine, guanine, ATGC. Okay, so these regions are called recognition sequences or the recognition sites and are randomly distributed throughout the DNA. Different bacterial species make restriction enzymes that recognize different nucleotide sequences. So ends of the restriction fragments are the blunt ends and sticky ends. Blunt ends are the nothing but the some restriction enzymes cut DNA at opposite base. They cleave blunt end DNA fragments. These blunt ended fragments can be joined to any other DNA fragment with blunt ends. Enzyme useful for certain types of DNA cloning experiments. Sticky ends, most restriction enzymes make staggered cuts. Staggered cuts produce a single stranded sticky ends. Sticky ends are very useful. DNA fragments with the complementary sticky ends can be combined to create new molecule which allows the certain and manipulation of DNA sequences from different sources. So isogenomers and the neoschizomers, restriction enzymes that have the same recognition sequences as well as the same cleavage site are isogeomers. Restriction enzymes that have the same recognition sequence but cleave the DNA at different site within that sequences. Okay, so this is the difference between the isogeomers and the neogeomers. These types are categorization based on their composition, enzymes cofactor requirement, the nature of their target sequences, position of their DNA cleavage site relative to the target sequence. There is a great deal of variation in restriction sites even within a species. Although these variations do not have phenotypic expression beyond the base sequences themselves, the variants can be considered molecular alleles and they can be detected with the sequencing techniques. As such, They can be used in mapping studies, similar to the way through genes with non-phenotypic effects can be used, but skipping the breeding steps and going straight to the molecules. These molecular alleles are a type of the molecular marker as they can be detected and located with labeled probes. So, ordinary nucleases are endonucleases or exonucleases. The forum gives the DNA backbone between two nucleotides, that is, it leaves the double stranded DNA at any point except the ends, but it involves only one strand of the duplex. The latter remove or digest one nucleotide at a time, starting from 5' or 3' end of a DNA strand. The restriction endonucleases cleave only at specific regions in a particular DNA so that discrete and defined fragments are obtained at the end of total digestion. The name restriction endonucleases originated from an observation of a system of restriction of the growth of the page lambda in particular strains of the E. coli host. So, most restriction enzymes recognize only one short base sequence in a DNA molecule and make two single strand breaks, one in each strand, generating 3' OH and 5' P, P means phosphate groups at each position. The sequences recognized by restriction enzymes are often palindromes, means that is inverted repetition sequences which are symmetrical. Note, each enzyme recognized and cuts specific DNA sequences. For example, BAMH first recognizes the double-stranded sequences. Restriction enzymes can be cut DNA in two ways to generate a blend end. Blend end means cut precisely at opposite sides, that is a H pole, and staggered ends. Staggered ends means sticky ends. Cut at asymmetrical position, example echo or one. A short single-stranded overhangs at each end. A large number of restriction enzymes have been identified and classified into three categories on the basis of their site of cleavage: type 1, 2, 3. Sometime in many other texts it is the four types: type 1, 2, 3 and the 4. Restriction enzymes have three important features, means they make breaks in palindromic sequences. The breaks are usually not directly opposite to one another. The enzymes generate DNA fragments with complementary ends. So these are the three important features of the enzymes. So function of the restriction enzyme or restriction enzyme generally found in bacteria and archaea and provide a defense mechanism against invading viruses. So restriction enzymes are enzymes that cut DNA at or near specific recognition nucleotide sequences known as restriction sites. Isolated restriction enzymes are used to manipulate DNA for different scientific applications and are an important tool for recombinant DNA technology. They are used to assist insertion of genes into plasmid vectors during gene cloning and protein production experiments. Restriction enzyme can also be used to distinguish gene alleles by specifically recognizing single base changes in DNA. They are used to digest genomic DNA for gene alleles analysis by Southern blot. To summarize, most restriction enzymes are specific to a single restriction site. Restriction sites are recognized no matter where the DNA came from. The number of cuts in an organism's DNA made by a particular restriction enzyme is determined by the number of restriction sites specific to that enzyme in that organism's DNA. A fragment of the DNA produced by a pair of adjacent cuts is called restriction fragment, and the site is the restriction site. A particular restriction enzyme will typically cut an organism DNA into many pieces from several thousand to more than a million. Thank you. In the next class or this slide, I am going to explain the types of the restriction enzyme.